Hello everyone. This is Jihan University of The Hague, Medical Microbiology Department, 4th year, Water and Soil Microbiology Practical. This is Lab 3. Today is the detection of degradation of starch by soil microorganisms. In the soil is very rich and complex composition, and this is a varieties of microorganisms in the soil, all of types of microorganisms, and have a lot of roles and importance in the soil. If you remember in the theory you have in this theory lecture, humus. Humus is very complex in the soil. So it's composed of organic compound, the carbon and nitrogen, with combination is lignoprotein, of course, and inorganic as well, and inorganic salts like phosphate and others. So it is this composition, of course, depending on a lot of factors. Today, in the practical, we will not focus on the theory part. Just some tips before we go to the practical, of course. So, and it is the factors are constitution of dead and living plants and animal cells, in which enter to the compound of humus, and the interconversion of material by various types of microorganisms in soil conditions like temperature, soil reaction, aeration, and moisture, of course. Look, this is types of humus, and they, of course, are different in color and structure and moisture as well. How the humus formed, the importance, sources of a humus are plant residues and stable manure, green manures, and artificial composts, of course. The sugars, starches, easily degraded, followed by some hemicellulose and proteins and celluloses, of course. If the material is rich in nitrogen and darkening takes place, so the process of decomposition becomes slower, and the residual mass turns brown to black, and this is why the soil and have this color, of course. Here, as you have seen, of course, with the microorganisms in the humus and the worm, in the soil. So you know that these worm have a lot of importance in the soil. We have a lot of functions, of course, of the humus. It improves the physical condition of the soil and increases the water holding capacity of soil, serves as a source of energy, and the humus possesses cation exchange capacity and ability to absorb water, and of course increase the fertility of soil. Also, the humus is a great storehouses of carbon and nitrogen and salts, influences and chemical properties of the soil, and the plays as a very important role in soil-plant relationship and texture and structure of the soil. In soil, many microorganisms have the ability to produce enzyme like tyrosinase and other enzymes. So this used to oxidize the amino acid tyrosine in soil. So this is located in the environmental of the microorganisms as a single source of carbon and energy, of course. This convert to melanin according to the according to a specific equation, of course. The microorganisms possess many enzymatic systems, natural or stimulating. You can do this by stimulating or natural. So decomposition is depend on this reaction and this importance of enzymes as well. So one of the enzyme which the microorganisms produce, and so this practical lab today is the degradation of starch. So starch, as you know, contains and composed of a polymer of glucose. Starch it's a polymer of glucose, and the microorganisms produce enzymes, is amylase, alpha or beta and beta amylase. So this will break down the starch and do degradation of starch into consisting of starch into units, which is glucose. How can I detect the starch hydrolysis by microorganisms in the soil? Look, if I have the 1 gram of soil and then inoculated in the distilled water, for example, 1 gram to 9 milli or to 99 milli of distilled water, in the and then do the dilution, 4 or 5 dilution, and then have 0.1 milliliter or 0.5 milliliter on the nitrate, on the starch agar, nitrate agar with starch. So the colony will, after incubation in the 30 centigrade and around 4 to 5 days, the colony, of course, will appear on the in the petri dishes in a starch agar, and then around the colony you will find like this. You will find like a zone, so a zone around the colonies, and we can detect it by iodine solution. So will appear like this, and then reaction it is between starch and iodine will give like this, and the other sites on the petri dishes no starch hydrolysis. We can detect the starch hydrolysis by iodine. This is the dilution, as I said, and the procedure as I now said for you, 1 gram to 9 milliliter, and the dilution. And look, this is the zone around the colony, and this is by iodine you're going to detect it. So this is the detection of starch degradation and starch hydrolysis, of course. This is the continuing of the procedure, and thank you for your listening.